Triclosan is a common ingredient used in antibacterial soaps. It is found in many products such as the ones listed on the slide. Recently, scientists have documented harmful effects on various species resulting from a high concentration of triclosan. These effects included nerve damage, hormonal dysfunction, and skin abrasion. Widespread use of triclosan in everyday products predicts future environmental concerns, including the contamination of the aquatic ecosystem in the Chesapeake Bay. This experiment targeted the chemical's effect on the larval blue crab. Factors tested included mortality rate and physical growth. Physical growth is measured by a term called molting, which describes a crab's transition between life stages. So this is a picture of the Chesapeake Bay. It's one of the largest estuary systems in the world, and its many inlets provide diverse aquaculture. Different well plates were set up with various concentrations of a dilution of triclosan in 30 ppt, or parts per thousand, water. The concentrations ranged from 0.1% triclosan to 1 times 10 to the minus 8%. In each cell, 2 Z8 blue crab larvae were exposed to the treatment. The crabs were fed 10 to 15 fresh Artemia, or baby brine shrimp, and the acidity level of the water was kept at approximately 8, which is standard for seawater. To the left is a picture of Artemia. We use it to feed the crabs. And over there is a picture of a standard Zoea crab in its late larval stage. This is a picture of the life cycle of the Chesapeake Bay blue crab. Our experiments primarily targeted the crab at the Zoea, Megalopy, and first crab stages. After 2 days, results were taken from the 0.1% triclosan treatment. As you can see, there was a 100% mortality rate among the crabs between the 2 days. The 1 times 10 to the minus 8% treatment, however, experienced better results after an 8-day cycle, as only 16 out of 48 crabs died. This graph depicts the molting rate of the crabs in the 1 times 10 to the minus 8% triclosan treatment. As shown, the crabs seemed to molt to the megalopy stage with a prolonged exposure to the treatment, and one crab, by the 9th day, reached its first crab stage. The results from this experiment illustrated a 100% mortality rate among the crabs until a concentration of about 1 times 10 to the minus 5% triclosan. Also, molts increased with a decrease in triclosan concentration. After a concentration of 1 times 10 to the minus 4%, 2 crabs were placed in each cell. An observation from this adjustment was cannibalism between the 2 larvae. Overall, the crab larvae did not respond well to higher or medium-range concentrations of triclosan. At lower concentrations, they seemed to respond with a higher survival rate, and at 10 to the minus 8%, 1 larvae molted all the way to crab 1 stage. However, there are many variables in this experiment, such as the feeding of the larvae and the cleaning and resetting of the water for the well plates, so that future examination of the effects of this chemical on crab growth is necessary. My second experiment involved the impact of different salinity levels on larval crab growth. The measurement of salinity level is the determination of the salt-to-water ratio within a specified amount of water. In this experiment, the salinity level was measured in ppt, or parts per thousand, and most labs keep larval blue crabs in water of about 30 ppt. The objective of this experiment was to determine the lowest salinity level in which Z8 blue crabs can survive and molt. By finding a lower salinity level for the crabs to live, it will hopefully reduce the cost of maintaining a saltwater aqua system. This is a picture of a typical tank at my lab. Its circular shape provides easy reproduction of a current in water. So, 2 experiments were set up using 2 different tanks of Z8 crabs. For tank 810, 8 well plates were set up with salinity levels of 15, 20, and 25 ppt with 1 control. For tank B2, 4 well plates were set up with the same range as tank 810. Both experiments contained 1 Z8 crab per cell and were fed with 10 to 15 fresh Artemia. This chart depicts the survival percentages for the respective treatments of tank 810. 25 PPT yielded the best results, with 25% of the crabs exposed to that treatment surviving. The crabs in the 15 PPT treatment, however, did not last the extent of the experiment. A noted observation was the poor survival rate of the control. The second treatment provided better survival rates in general, with a small percentage of the crabs exposed to the 15 PPT water surviving. Once again, the 25 PPT treatment yielded the highest survival rate. This next chart compares the two tanks' molting progress. As shown on the left, a higher percentage of the crabs in tank B2 for 25 PPT reached the first crab stage or higher, while none in the 30 PPT or 15 PPT treatments molted that far. In tank A10, once again, the 25 PPT treatment provided the most molt to a C1 stage or higher. Overall, 25 PPT provided the best results with the most molts to C1 stage and had the highest survival percentage. Tank 810's lower survival rate than tank B2 was genetically confirmed at the lab in Piney Point, Maryland. Future experiments must be performed with a smaller increment and salinity level change to calculate the optimum level for crab growth. Both experiments concerned environmental factors affecting the blue crab population. As proven in the past, a slight decrease in population of one species can alter the ecosystem of the enclosed environment. I therefore learned not only of the biology and cycles of the blue crab, but also of the importance of environmental protection and conservation. I would like to thank my mentors, Dr. Suk Chung and Dr. Odie Moore. I would also like to thank my parents, Dr. Edward and Mrs. Faith Wolf, Mrs. Sephora Shore, Mrs. Susan Celeste, and Mr. Jerry Burgess. I also like to thank my grandmother from traveling from New York on Amtrak. Next, I would like to introduce the principal of Bethel High School, Rabbi Frank.